Hello and welcome to the Carbohydrate Chronicles, a podcast about glycoscience and glycoscientists. In this show, via a series of interviews, we explore the world of carbohydrate science and innovation from a personal point of view. Your host today is Tamas Shahai, the member of Carbohydrate's scientific team. For decades, scientists lived in a paradigm that cyclodextrins are useful excipients and additives with numerous applications, and that was mainly it. Pioneers came then and broke these chains, releasing the true potential of these molecules. First, Sugammadex was discovered, which is now a blockbuster drug. Later, long-known cyclodextrins were repurposed as active ingredients for various therapeutic indications. Some of these developments are now in clinical trials for rare genetic indications or Alzheimer's disease. In parallel, research started to flourish both in the academia and industry, and companies like Cyclarity Therapeutics were incorporated, focusing now on cyclodextrin-based drug discovery and development. Today, our guest is Dr. Matthew Oki O'Connor, founder and co-CEO of Cyclarity Therapeutics, whom I have the pleasure to know for several years now, from the very beginning of the Cyclarity story that we will learn a lot about today. Welcome, Oki. It's great to have you on the show.
Thank you so much, Tomas. It's a pleasure and an honor to be here.
Oki is a true longevity barrier who dedicated all his professional career to this research area. After finishing his studies, he almost immediately joined Sense Research Foundation as head of research to develop, promote, and ensure widespread access to therapies and cure and prevent diseases and disabilities of aging, which position he left for Cyclarity eventually. He's also a board member of Longevity Biotech Fellowship and a senior researcher of VitaDAO, a decentralized collective funding initiative for early-stage longevity research. Oki, you seem to have a very widespread activity within the longevity space. How do you balance all these activities?
Well, it's just the main focus of my career and my life. So obviously, 99% of my work is on my company, running my company. Some spare time working to support nonprofit and advocacy areas of longevity research and policy and education. So it's a pleasure to be able to do some of those volunteer efforts. But of course, running a startup biotech pharmaceutical company is pretty all encompassing.
With all these voluntary activities, is this some kind of a payback a little bit to, you have been on the other side of the research at Sense Foundation, and you know how important it is to support new ideas, new concepts. Is this to some extent how you pay the debt?
Yeah. I guess I haven't really thought about it that way exactly, but yes, I think that's a good way to put it, Tomas, because there's no world I think in which we have only private science, private research, and I don't think there's a world in which you have only public research. There really is a back and forth. A lot of the technology that Cyclarity is based on was invented in the nonprofit space. So there's a real need for that side of things. And then, the other kinds of stuff, the advocacy, the education, training new researchers, scientists, students is really key to, keep bringing on a fresh crop of people, of ideas, of workers to help spread the ranks.
What made you, if you go back to your childhood, what made you choose science in the very beginning?
I guess I'm the first scientist in my family, but both of my parents were computer programmers. So, math and technology was certainly encouraged in my household and in my education. And I guess I have a debt to my father, who from a super young age really tried to instill ideas in me of, scientists being the heroes and the superstars of the world rather than lionizing, rock stars or sports stars and such like that. So buying me, math games and buying me, books. Like I had a comic book about Albert Einstein when I was 6 years old or something like that. And so, growing up in that kind of environment certainly paved the way for me to be able to become a scientist.
So computer programmers, were they trying to get you into the Silicon Valley, or
No,
it was
not at
completely?
all. They never pushed me. In fact, I'm not a technophobe at all, but I couldn't code my way out of a wet paper bag. They never taught me anything about coding.
Okay, I understand now your connection to natural sciences, but what made you pick biology or biochemistry? I'm not sure what's the best way to put your main interest.
Well, the reason I decided to become a biologist was because I decided when I was 16 years old that I wanted to work on aging and that I wanted to do something about longevity. And that was my entire motivation for first majoring in the natural sciences and then, focusing on biology, biochemistry, molecular biology, cell biology, because I felt, and I think it was essentially the right decision, that aging at the molecular, biochemical, and cellular level is really the level at which we should be thinking about it and the level at which we should be intervening if we want to come up with therapies that target the diseases and disabilities of aging.
And you figured it out at the age of 16? That's a
Well,
very
I couldn't have articulated it quite so well, but, I was reading the, sort of early stuff that I could get my hands on about, telomeres, which are the ends of the chromosomes that shorten with every cell division and shorten in most of our cells and most of our tissues. And replicative senescence had been discovered in the late 60s. So I could read about that. I read Len Hayflick's book How and Why We Age when I was a teenager. So that early amounts of stuff that I could find and get my hands on is what guided me to make those decisions. And then, of course, as I went through my schooling and started learning how to read journals and such like that, that helped focus me even more.
It's very rare that young teenagers keep focused on their dream for such a long period. After spending now quite a long time in this research area, what is it that motivates you most in it? What do you enjoy most in longevity research?
Well, for me, it really is about the goal and about the potential to actually have the chance to bring therapies to large numbers of people and be able to impact chronic disease at a massive scale. You have a chance to actually do something. So I feel really lucky to get up every morning and do exactly what I dreamed of doing ever since I was 16 years old.
It's really great to hear when people are so passionate and happy about what they're doing, and then at the same time, they have a very important mission that they're working on. If we jump a few years ahead, you were already at Sense when you came across first with cyclodextrins, or what's your first memory with these molecules?
Yeah, that's exactly right. I didn't sort of grow up, if you will, with cyclodextrins at all. I didn't learn about them at all during my training. I ended up getting exposed to and attracted to cyclodextrins because we had a project at the foundation that I had oversight for. We had a longstanding project to try to target oxidized cholesterol that accumulates with age in various cells and tissues. And my interest in that actually went back to graduate school, but had nothing to do with cyclodextrins. When I saw one of my mentors, who was the founder of the foundation that you were just talking about, give a talk about how the accumulation of oxidized cholesterols inside of macrophages turns them into foam cells, which are these cells that are supposed to go in and eat up all the junk in an atherosclerotic plaque in the arteries. They're supposed to go and clean it up, but instead, when they eat too much oxidized cholesterol, it shuts down their lysosomes, which is the garbage disposal unit of all cells, and they end up ballooning up into these nasty cells called foam cells that become part of the problem rather than the solution. So But we had this sort of fancy enzyme engineering approach, which was very cool, but also difficult to develop and translate into a therapy. And so I started casting about for what's a hack? What's something easy that we could do that we could just sort of drop in as something that we could develop relatively quickly and get to clinic. That's what led me to to cyclodextrins, which you had all of this work being published about the various cholesterol binding properties of beta cyclodextrins, especially. And, diving back into that old literature is what started generating these ideas for me, how we might be able to engineer new cyclodextrins to do exactly what we wanted to do in a customized fashion.
So you studied the experiments since, and when they were promising, you decided to spin out and create a company of your own, focusing specifically of this project with your co-founder?
Yeah, exactly. I started off with a tiny budget that I begged for from, our funding committee, and I got like one intern to work on it for, I don't know, half a year, and everything worked. It's one of those things where, everything you try works the first time, and got really lucky in that regard. And, then I went and begged for more money and got a bigger budget for it, and was able to, hire some people and started spending more and more of my time. And eventually we invented some new technology, the dimer technology, together with Cyclolab. We put this together. We tested the first dimers. They worked almost magically well. I still remember the first time that I tested them, and I could just see by eye that they were working, and gathered a lot of data and wrote the first patent. And from pretty much from that moment forward, when we made the first dimers and I could see how well they were working, I knew from that moment that this needed to be a company, and that I was going to try to make it into a therapy.
That's a very, very nice and stimulating story for academic researchers. Many people in the academic field dream about these big discoveries that they can eventually spin out and live for later. For you, was it always a dream to become an entrepreneur?
More by chance. I wouldn't say that I am a entrepreneur necessarily by nature. If I think a problem can or should be solved in this or that sector, I'm happy to do it in academia, nonprofit. Drugs are often conceived of, and the core of them is often invented in academia, but in terms of actually developing drugs, that happens in companies. So that was the path that it needed to take, and I was happy to throw myself into it. And I guess I feel like I have a bit of a knack for entrepreneurism. It suits me well. It's fun. It's intense. I think this is a common theme among first-time entrepreneurs, is how intense it is, and how much harder and faster you end up working. And what really, I think, took me by surprise, I knew that I had a lot to learn about pharmaceutical development, but I just had no idea how many facets of various administrative and financial and legal and management skills that I was going to need to learn very quickly on the fly. So that's been a challenge, but it's also just been kind of the time of my life.
I can also imagine that you need to give up a lot. What is it that you enjoy most in your current position as a CEO? And don't you miss doing more research?
I have a great co-founder, Mike Cope, who he was the CEO of the foundation, who came with me, and he's not a scientist, he's a lawyer by training. So he can certainly help out with a lot of the administrative legal. He's a contract wizard. which I think is really important. Do I miss, being closer to the scientists? I meet with my bench team. I meet with them every week, and my door is always open. And I feel like if I was, Captain Adama, that they're my fighter pilots of my squad, right? I value them tremendously as the scientist. My scientists are my all stars that I love and need to take care of.
And you do most of that support to keep them motivated as their leader, I'm sure. How about activities outside the lab? Do you switch off easily?
It's hard, I admit. I close my eyes and and I'm worried about funding, or I'm worried about experiments that are running, and I'm worried about contractors. But it's important to have a life outside of science. And so throwing yourself into something that kind of forces you to think about nothing but that, I think is a good way to switch off, as you were saying. So I'm a lifelong martial artist. I've done martial arts since I was 6 years old. I belong to an MMA gym. I do kickboxing and jujitsu. And mixed martial arts. Yeah, there when you're like wrestling around with a partner or an opponent, you're really only in the moment, and that's a nice kind of way to keep myself physically and mentally, sharp in a different way, and work off some of my stress in a healthy way.
It's certainly not a time to worry about funding if someone is trying to.
Somebody's trying to choke you. Yeah,
yeah.
You're not so worried about your funding problems.
Let's talk a little bit about now Cyclarity Therapeutics. Where are you with your development now? What do you focus on currently, and what's next?
Yeah, so we have this dimer technology that we've pioneered and focused on, where we take two cyclodextrins and stick them together in a head-to-head conformation. And the basic idea is that we can create a cyclodextrin that, under the right circumstances, can increase its affinity for its target by, let's say, 1,000-fold. We have this platform technology that we've developed in collaboration with our collaborators at the University of Santiago de Compostela on how to simulate cyclodextrins computationally. So that's sort of a whole mini department of our company that comes up with the concepts for prototypes that we can synthesize. What we spend most of our money and time on is developing our lead drug candidate, which right now is, just has a code name UDP-003. And what it does is it encapsulates a specific oxidized form of cholesterol that, like I said before, accumulates in various cells and tissues with age, especially in these foam cells that accumulate in plaque that builds up in your arteries. And so we're very far along in developing this first drug for clinical trials. We've scaled it up, we've manufactured it, we've made the material that is going to go into people. And we're, doing the late stage IND, IMPD enabling work, which is, all the safety and pharmacology tests that the regulators insist that you do before you do your first in human trials. And we're aiming to start our first in human trials right around the end of this year, the beginning, probably, being probably a bit optimistic, probably more like the very beginning of next year.
Wow, that's very exciting. That I'm a huge fan of new cyclodextrins, and I think that this is the first new one used in humans for a very long time. When you talk about the technology, you seem to use the concept as a platform. So I was wondering if you have ideas to use UDP-003 for other indications. or you have other problems in mind for which you would design new compounds on the same concept?
Yes. What I want to say to people who are interested in the pharmaceutical audience is that when you target a basic mechanism of aging for a disease, you're going to have multiple indications for sure that you'll be able to use it on. And so, for example, the largest government grant that we've gotten for the company was actually to work on Alzheimer's disease. That's a potential indication. Macular degeneration is a potential indication. Stroke recovery is a potential indication. And there may be indications that we haven't discovered yet. Non-alcoholic fatty liver disease, basically age-related liver failure. I actually wrote a review article that we published in 2020 about oxidized cholesterol and its chemistry and its biology. And, we go through all of the diseases that it's implicated in. Niemann-Pick type C disease is an orphan disease that hyperaccumulates. It's a cholesterol transport disorder that hyperaccumulates oxidized cholesterol that I think our drug would be effective against. So I think there's many potential indications for our lead drug. You asked about our technology and our platform, and on that side, I just think there's such enormous potential. It almost hurts my brain to think about how we can, how could we possibly, exploit all of the potential of our technology? What I'm most interested in and what my company is most focused on is the pharmaceutical API applications. And I don't understand. There are other companies like mine that are developing cyclodextrins as APIs. I don't understand how big pharma isn't investing in this massively. There's huge opportunity here, and I want to make the case that cyclodextrins, the way that you can engineer them, they're sort of the perfect compromise that are halfway in between small molecule drugs and therapeutic antibodies. And so I think that there's just enormous potential there for many drugs on the market. And the regulatory path that Sugammadex blazed and the excipients that are approved that cyclodextrins can be used for, the combination of those means that regulators are pretty comfortable. They know, every regulator that we talk to at FDA or MHRA in the UK that we've done a lot of meetings at, they're all familiar with cyclodextrins now. There's a path to follow there for pharmaceutical development. Yeah, and I didn't even touch on yet, about the non-pharmaceutical applications of cyclodextrins, which, if you want to get into, we can try to talk about that.
I truly share your vision and optimism about the bright future of cyclodextrins.
We wrote a grant that, about using part of which would be using our cyclodextrins that we can develop in silico, in the simulations for, environmental remediation and things like that, which I know you're familiar with. Yeah. That kind of thing, materials development, environmental stuff. My dream is that when we get a little bit more popular and famous, is that those people will start coming to us and offering to partner with us. So for your particular audience, I really want to encourage people who, take a look at our technology, use your imagination, and come to me with ideas of stuff that you think you can use our dimers for, and let's work together on stuff that we can build new cyclodextrins together for your applications, and come and take them from us, license them, do some sort of joint venture or something together, and hopefully our technology can spread in that way, because as biomedical researchers, we're not qualified to develop environmental remediation or some super materials and such, like all the cool, weird stuff that you can do with cyclodextrins that, I think our technology could be great for.
I really like this 2 feet on the ground approach, that you realize that, of course, you have capabilities and priorities, but still are very open to share what you have discovered with other research fields and understand the importance of those collaborations. One last question, Oki, a non-scientific one. If you could have dinner with any historical figure or celebrity that are alive, who would it be and why?
I think my favorite one will be Paul Erdős. Am I saying his name correctly?
Close enough, close enough.
When I was a graduate student, I read a biography about his life called the Man Who Loved Only Numbers. And I completely fell in love with this crazy mathematician who just derived so much joy from working on mathematics from dawn till midnight, like every day of his life. And I briefly had this fantasy of trying to imitate him and basically becoming a permanent postdoc and going from lab to lab and being sort of a vagabond who just wandered the world working on aging science and all the most interesting and coolest labs around the world. I think he is one of my heroes, and I would love to sit down with him and pick his brain and I don't know, try to learn something about how his genius worked and how he was able to be so incredibly prolific and productive.
Speak the Hungarian. I really loved it. Okay. I hope you enjoyed the most recent episode of Carbohydrate Chronicles today, delivered to you by Carbohydrate. We talked with Matthew Oki O'Connor, founder and CEO of Cyclarity Therapeutics, a bright and new star on the sky on cyclodextrin-based drug development. If I wanted to capture Oki's main message, I could probably say that being passionate and persistent to our long-term goals pays off, and working on big, massive problems of humanity is still an important challenge. And even more importantly, that I can only echo his views that there are huge opportunities in cyclodextrin-based APIs and other applications of cyclodextrins. So investors and big pharma, you should look more into this opportunity in much more detail and help us bring new compounds to the market. Thank you for being here with us, Oki, today. This was a fascinating discussion, and I learned a new side of you, which I'm very grateful for.
Thanks, Tomas. It was a pleasure, and I had a lot of fun.
Thank you very much for listening to the most recent episode of Carbohydrate Chronicles. Next week, we'll talk with Barbara Rikiki, Associate Professor of Chemistry at the University of Florence, expert in carbon and glycan-based nanostructured materials, who is just about to set up an international training network on novel glycomimetics to treat cancer. Your host was Tomas Shahida from Carbohydrate. I hope you'll join me in our next session too. See you in 1 week. Goodbye.